Now in this class we are going to discuss microbes in human welfare. So the microbes are present in everywhere. So in aquatic region and terrestrial region. So entire environment, anywhere it can be find these microbes. Soil region also. So how it is, these microbes are here used in this household products. So see here different household products, curd and dough and toddy and also cheese. These are the different kinds of household products. So curd, how it is made? So for the curd preparation, here participating that bacteria, it is a lactic acid bacteria. So this lactic acid bacteria to release the acids and also coagulate and also partially digest the milk proteins. And next, in this, it is this lactic acid bacteria. It increases the nutritional quality. What is that? Nutritional quality by increasing the vitamin B12 in the stomach region. And also one more, it play a major role checking the disease caused microbes. It checking the disease caused microbes. Next, second one, it is the dough. So here now we are making at home for preparation of it is idli and dosa material. So for this dough material, whatever the material it can be used, you can put in the inside. That means you can put in whatever you can mix all these materials. So what will that you can, we are stored at nearly about it is 4 to 5 hours. What will happen? It is the dough material, it can be fermented. The dough material, it can be fermented, so it can be forming holes. That means it releasing the carbon dioxide. That means it can be the dough material, it can form after that few hours later. Puffiness is can be formed. That means here that is a dough material. So this dough material is used. Okay, it is preparing the cakes, particularly in the bakeries. So that means here in this the dough preparation also. That means during the bakeries industries use the yeast. So which kind of what kind of yeast is used means Saccharomyces cerevisiae. The Saccharomyces cerevisiae is used, so it is preparing the cakes. So that's why this is we call this yeast, we call it is a baker's yeast. And also this yeast is used mainly in the production of some of the beverages such as it is alcohols. Next here the toddy. So actually it is the toddy is the traditional drink, that means natural daru we call. So this the fermenting sap. So this is the fermented sap material. It is comes from the plants. This fermented sap is comes from the plants such as it is the palm plants. Okay, palm plants. So suppose for palm plants and also phonix dactylifera. These are the examples. These are comes to this toddy traditional drink. And also cheese also. So here we are seeing say this one. It is the Swiss cheese. The Swiss cheese, it can be producing the carbon dioxide. The Swiss cheese, it is a large holes from that here production of it is the carbon dioxide. So the Swiss cheese formation here use the bacteria, it is the Propionibacterium shermani. So next one more cheese is the Roquefort cheese. This is the ripened cheese. The ripened cheese mainly it is the Roquefort cheese grow on the region, it can be grown in grow the fungi. Okay, the Roquefort cheese on this grow the fungi materials. And also this Roquefort cheese is comes from, it is a Penicillium roqueforti. So these are the household products. And also some of the soy products also it is used as a food material in some other countries. Next industrial products. Industrial products means what are the industrial product, different organism, how it is used. So the different organism names they can ask in the NEET and also CASIC examinations. So the main microorganisms are very important. Suppose you can see here yeast. Yeast is generally hybridized, it is a baker's yeast. This is the baker's yeast. The yeast is used to preparing the cakes and bakes. So that's what we call it is the baker's yeast. Next Alexander. Okay, so antibiotics word. So anti means against. Okay, so this antibiotics word term it is coined by scientist Waxman. So the first antibiotic is discovered by scientist Alexander Fleming. So this Alexander Fleming, first of all, they are observing, they are conducting experiments on Staphylococcus bacteria, Staphylococcus. So in the Staphylococcus bacteria, it can be observed. So the surrounding some unwanted, it is the fungal material is growing. So that is contaminated to this bacteria. So the from this fungi, isolated the material. So this fungi, it is look like a brush-like appearance. That's what is this fungi? After that, it was identified that is called, it is a penicillium. So from this penicillium, isolated the component that is called, it is a penicillium. Sorry, from the penicillium, producing the component, it is a penicillin. So the penicillium species, there are different types of species are there. So the penicillium notatum to producing the antibiotics such as it is penicillin. So during the Second World War time, that can be used is the penicillin product only. Next, this penicillin mainly we have to treat plague and also whooping cough. What is this one? Whooping cough. That means generally we call it as a colic calci. And diphtheria. That means a gall goti and leprosy kushtrobose. So this is the penicillin is used for different diseases purpose. And different microbes are they are producing the different products. Suppose here Aspergillus niger. So here Aspergillus niger, it can be production of it is a citric acid and Acetobacter acetic. This Acetobacter acetic can be producing the acetic acid and Clostridium butylicum. So the Clostridium butylicum, it can be producing the it is a butyric acid and Lactobacillus lactobacillus to producing the lactic acid. And here yeast is producing the ethanol. Okay, they are different kinds of products. So what are the organisms, in which organism the production of particular product, you can see these are the most of the examples. So these are very important. So these number of organisms, what are the organisms are present, those organisms are what produce the product based upon number of questions we will ask. Okay, next see here enzymes. Enzymes means here see lipases, proteases and pectinases and streptokinases. So here lipases. So lipases means it is mainly used in the detergent formulations. Okay, to removing the cloths, that means oil clearing. That means what are the oils are present in the cloths, that it can be removing these lipases. That means the lipase mainly it can be used in the detergent formulations to clearing the oils from the cloths. Next, proteases and pectinases. These proteases and pectinases, it is mainly used to clearing the juices, clearing the some of the fruit juices. Suppose in shopping malls, so the fruit bottles you can see very clear because of here the presence of it is the proteinases and pectinases. So the clearing the juices purpose that can be used. So these enzymes are used. Streptokinase. So it is the Streptococcus bacteria. From that it can be producing this enzyme. So it is mainly, so this mainly used as for clot. Generally which purpose are used, it is clot buster. So a person who is suffered the myocardial infections, so that is for that purpose it can be used as a streptokinase. Its main function is that remove the clots in the blood. So that's it is the, this is generally called it is a clot buster. Next, bioactive molecules. So what are the bioactive molecules? Here the bioactive molecules are cyclosporin A and statins. Okay, statins. So here cyclosporin A. The cyclosporin A comes from organism, it is a Trichoderma polysporum. Trichoderma polysporum. Trichoderma means it is a fungal organism. Here what are the chemical, any component comes from the component name also will give, organism name will give. So what are the organism belongs to the group sometimes. Okay, the organism is belongs to the group, such a questions also will ask. So that's why the cyclosporin A, it is comes from the bioactive molecule, that is Trichoderma polysporum. So here the Trichoderma, it is main we call it is a fungal organisms. And also statins. So the statins it is actually it is comes from the yeast organism. So it is Monoscus purpureus. Monoscus purpureus. Okay. So which purpose are used? So here the blood cholesterol lowering agent. Okay. These statins are used for blood cholesterol lowering agent. Okay, these are the bioactive molecules. Next we can say is the sewage treatment. So here the sewage treatments are, you can say the two methods are present. It is the primary treatment or secondary treatment. So primary treatment is a biological process, secondary treatment, sorry, primary treatment is a physiological process and also physical process, and a secondary treatment is a biological process. Okay, so in the sewage treatment, what are the waste material is used? There are different techniques. Okay, different techniques are used. So what are the floating debris and sedimentation process later? It is the different kinds of, it is in the primary treatment is effluents. Okay, effluents are formed. These effluents are transferred into in the secondary treatment large aeration tank. In the large aeration tank, the vigorously growth of it is the bacterial flux. What is the what is the flux? Not bacterial flux. Vigorous growth of flux. Flux means actually it is the bacteria and also fungal material together. It can be growing. So that's what we call it is the flux. So the flux, whenever it is flux, it can be growing, vigorously growing. So after that, it is whenever it is in the primary settling tank, it is BOD is reduced. BOD is reduced. So BOD is reduced means automatically pollutants can be comes down. So whenever the BOD is reduced, the some amount of it is activated sludge is pumped into another tank. Is a second another tank, that is anaerobic sludge digester. Remaining the anaerobic sludge digester, it containing that, it containing the anaerobic bacteria. In the anaerobic bacteria and the anaerobic sludge digester, some sediment. So this sediment is present. So this anaerobic bacteria digest the aerobic bacteria. So as a result, it finally producing the biogas such as it is methane and carbon dioxide and H2. Okay, this is the sewage treatment. So next, in this next biogas plant. The biogas plant is made the concrete. It is nearly about it is a 10 to 15 feet is present. So then what are the waste material that can be put into this biogas plant, and what is this one? It is what are the, it is a biogas production purpose. It is this biogas plant is used also, and the cow dung, what are the waste is present, that dung it is put into this concrete, this biogas plant. They are covered with some pepper, and also it can be this automatically what will happen after that, a few months, a few days later, it can be the biogas, it can be the pepper, it can be moves upper. So as a result, it is what are the biogas plant. So the gas is one outlet is present from the directly, it can be connected to some of the states, some of the countries, it is into the households also. So next here, biocontrol agents. So the biocontrol agents means nowadays it is chemicals are used into the fields. So how it is we are declining naturally means so nowadays are introducing the biocontrol agents. So it is used, the biocontrol agents is used to control the pest and insects. So naturally. So here see first one, the ladybird and dragonflies. Ladybird and dragonflies. So here ladybird and dragonflies get removing of aphids. Okay, the red and black markings of this ladybird and dragonflies get rid of it is these aphids. And next Bacillus thuringiensis. The Bacillus thuringiensis here production of it is the toxins. So whenever it is directly it can be injected, these beta toxins are injected into a crop field. So automatically the insect is ingest these toxins. So these inactive toxins, actually in the plant itself it is the inactive status present. So whenever this beta toxin ingest these insects, so inactive form it can be converted into active form. So automatically it is the insect it is in the epithelial cells, it can be formed the pores. So automatically it can be cracks, finally it leads to the death. So that's why it is a naturally it can be used with this biocontrol agent Bacillus thuringiensis. Next trichoderma. So this trichoderma also it is a fungal material, it is associated with the root system also. It is also can be controlled but it is also controlling and also used as a biocontrol agents. Baculoviruses. Baculoviruses means the insect. It is a baculoviruses or it is the insect specific. It is the narrow spectrumly used. Okay, so these baculoviruses, which insect is which insect is damage the crop. Such a insect collected that can prepare the components and also these are spread onto the such a field area. Automatically, what are the insects and also pests and also virus, everything it can be disappear. So that's why we call it is used the such a viruses, we call it as a bacilloviruses. Okay, these are the biofertilizers. Next, biofertilizers. So biofertilizers, what is meant by here? The biofertilizers. So biofertilizers means it increase the enrich of nutrient quality of soil. It enrich the nutrient quality of soil. So that's what we call it as the biofertilizers. So biofertilizers purpose is what are the organisms are main participating that. That means which organisms are used as the biofertilizers. Here suppose for example it is a bacteria and also cyanobacteria, okay, and also mycorrhiza. These are the used as a nowadays as the biofertilizers. Suppose here bacteria, rhizobium bacteria. So rhizobium it can be associated with the leguminous plants. So it can fix the nitrogen. Already you know that in first year, the rhizobium bacteria it can be associated with leguminous plant, legume plants, the roots of legume plants. It can fix the nitrogen. Okay, and azospirillum and azotobacter. Okay, azospirillum and azotobacter. It is a free living bacteria. It is free living bacteria. It is the increasing the nitrogen content in the soil. Okay, so this is the azospirillum and azotobacteria. Next, mycorrhiza. What is meant by here mycorrhiza? Myco means here fungi, rhiza means here root. Okay, so the mycorrhiza, suppose some members of example, the members of it is a glomus. The mycorrhiza is the members of it is a glomus. So mycorrhiza, what are the main functions? The main it is the mycorrhiza absorb the phosphorus. Mycorrhiza absorb the phosphorus and absorb the phosphorus. It is supplied to the plants. And also next the mycorrhiza, it is a plant resistant. What is this one? Plant resistant to rootworm pathogens. Next, it is the mycorrhiza can be tolerance to the abiotic stresses. Increase the tolerance to abiotic stresses such as salinity and also drought. Next, the mycorrhiza it is used to participate in the plant growth and also development. Plant growth and also development. Next, cyanobacteria. Cyanobacteria means it is present in the aquatic region and also terrestrial region. And also, what are the main cyanobacteria example? Nostoc, Anabina and Acylitoria. Okay, next, what will happen? What will you do this cyanobacteria? It can fix the nitrogen. It can fix the atmospheric nitrogen. Next, it is the cyanobacteria add the organic matter to the soil. And then it is increasing the soil fertility. Whenever it is spread, the farmers are this cyanobacteria spread into the field. So automatically it is the soil fertility can be increases. Okay, so these are the biofertilizers. So in the examination, some of the abbreviations also asked here. What is this LAB? Lactic acid bacteria. Here the dissolved oxygen. It is a BOD. Here what is meant by here? The BOD means it is a biochemical oxygen demand. Here STPs in the sewage treatment plant. Here the sewage treatment. Here the primary treatment, secondary treatment. Further it is nowadays the sewage it can be treated using the plants. Some of the most of the metropolitan cities in Hyderabad and Kolkata and also Delhi and also Bangalore such regions. What are the domestic water it can be present? That wastewater enter it can be sent into the particular areas. In such an area they are that means the governments they are constructing the plants. We call it is the sewage treatment plants. So that's we call it the sewage treatment plants. It can filter the water number of times. There are different method. What is the primary treatment, secondary treatment? One more it is the tertiary treatment. As a result, finally what is the water? This water again reutilized to the human welfare. Okay, that purpose that can be made. This is the sewage treatment plants. And GAP, that is Ganga Action Plan and Yamuna Action Plan and Indian Agriculture Research Institute and Khadi Village and Industries Commission and Tobacco Mosaic Virus and IPM means here it is integrated pest management. TM means Tobacco Mosaic Virus, KVIC. Okay, Khadi Village and Industries Commission. Here what is this one? Khadi Village Industries Commission. Okay, so TM Tobacco Mosaic Virus. IPM means Integrated Pest Management. Indian Agriculture Research Institute. IDRI means Indian Agriculture okay, Indian Agriculture Research Institute. So these are the microbes in human welfare. So number of it is microbes they can ask for examination. Next we are going to strategies of enhancement of food production. Next we are going to discuss it is the plant breeding. So here plant breeding, what is meant by here the plant breeding? The manipulation of different species to produce in the better yield, and also with such a plant species we are cultivated, you can see in the plant breeding. So what are the traits incorporated in the plant? What are the traits, that means characters incorporated into the crop plants? What will happen? So the traits, the desirable traits are can be incorporated. So increasing the crop yield, what will happen? It is increasing the crop yield. That means the yield, previous it is 20 bags will come, so nowadays it is maybe 50, 60, like it is the yield is increases. And also quality, it is improves the quality also. Whenever these desirable characters introduced into plant, what will happen? It is increasing the quality, and also increasing the tolerance to avoiding stresses, the salinity, and also cold and water and temperature, everything. This is increasing the tolerance to environmental stresses. Next, increase the resistance to pathogens. Such a crop varieties, it is increase the tolerance to the pathogen. That means pathogen means here virus and bacteria and also fungal material. And next, another character, increase the tolerance to insect pest also. Okay, so these are the main traits are incorporated into crop plants. So any one point of view, this traits in the introduced into any crop plant, what will be increases, that can be asked the questions. Next, plant breeding crops. So in the plant breeding crops, the new variety it can be developed. So that means what are the plant breeding steps? The new hybrid variety can be developed means what are the steps it can be utilized? So main we can say is the collection of variability. Here the collection of variability, evaluation and selection of parents, cross evaluation among the selected parents, and selection and testing of superior recombinants, and testing and release commercialization. Simply I am saying, you can say collection of variability. This is called it is a germplasm collection. So the collection of all the genes. So in the given crop we call it is the collection of variability. So next, evaluation of selected parents. Suppose two different varieties. What are the any desirable character is present? Such a desirable character plants varieties are we are selected. So those plants we are called the male and female character. Suppose one plant having protein is more, that we can select one of the pair, one pair, and another pair we can select it is a disease resistant. So this is the another pair. So both we are these two, one of them having protein character is high, another one it is the disease resistance is high. So the such a superior characters, two different plants are they are hybridized. So such a plants are selected. That's we call it as a parents. Next, cross hybridization among the selected parents. What you are selected that selected two plants are it is can be hybridized. What will happen? It can be produced in the hybrid. So after that we can select cross hybridization among the selected parents. So hybrid came. So the number of hybrid plants are produced, but every hybrid they are showing different yield. So how we are equality we are maintain. That means every plant. That means the plant it can be self-pollinated in several generations. So finally it leads to the homozygosity it can be formed. So that's why here how it is selection and testing of superior recombinant. The superior recombinant we can find how it is this self-pollinated several times, several generations, self-pollination several generations. Finally it leads to the homozygosity. So the which one is all the plants homozygosity form. So all the plants it is the same result will come out. So after that the scientists the testing and release commercialization. So what the plants are tested, and testing means that different areas are selected, 3 different zones are selected. So they are tested. So after that it is releases and we are giving the name and uses to the human welfare. That's it is the different steps. And improved varieties we can see. The green revolution it can be increased. The green revolution can be starts it is a 1960. Norman E. Borlaug, okay, sorry, M.S. Swaminathan. M.S. Swaminathan was the green revolutionist. So 1960 to 2005 it can be the improving the yielding and highly resistant varieties are developed. Suppose for example wheat. In wheat it is you can see it is during the 60 to 2000 time only here 11 million tons only present. So here achieving 11 million tons to 75 million tons between it is the yield is increases. and high yielding and disease resistant varieties of wheat. It is example Sonalia and Kalyansona varieties, and also the semi dwarf varieties. The semi dwarf varieties, such as it is the semi dwarf varieties IR8. It is the Indian Research Institute of in the Indian Research. So IRRI is developed. Okay, so next Teaching 81. So here Teaching 81. This is also one of the semi dwarf varieties. So it is arranged from it is the Taiwan region. Okay, and also the semi dwarf varieties. Indian Agriculture Research Institute IRI. So this IRI, Indian Agriculture Research Institute, can developing the semi dwarf rice varieties. Next, Jaya and Ratna. These are also one of the semi dwarf varieties. It will be developed in India only. So International Agriculture Research Institute. It is okay. So next, sugarcane. Sugarcane means actually it is, you can say here the Saccharum barbari. This is Saccharum barbari. It is the poor in sugar condition, poor in sugar produced. It is mainly growing in the North Indian region, and in India, that means South Indian region, it can be growing that it is the Saccharum obscurarum, so it can increase the sugar and a thick stem is present. So both it is these both are crossed with them, that means hybridized. As a result, what will happen? It is the here high yielding and also thick stem and such varieties it is developed. Okay, and also methods of breeding, that is the conventional breeding and methods of mutational breeding. So whenever the mutations are in this can be mutations are, it can be naturally, it can be occurs. So the mutagens are okay, the mutation, what is meant by mutations, what is meant by here mutation. So some of the mutations are, it can be naturally occurs, some of them we are provided, that means it is a chemically and also physically we are provided. So automatically it is that the plants, their character, it can be changes to production of it is a desirable feature also. So that's why here the mutational breeding, here the physical mutagens, ultraviolet radiations we are introduced into the plant, induced into the plant, that means inducing the mutations by using of these UV radiations. Automatically the new character can be produced. So it is alpha, beta, gamma, UV, alpha, beta, gamma. These are physical mutagens, and chemical mutagens means the calcium EMS, ethyl methyl sulfonate. So these are, it can be induced into the plant, so automatically arranging the new characters. Okay, and next one, it is the some of the one more here. Risk. One more, it is plant breeding for developing resistant to insect pests. Here main hairy leaves. Hairy leaves are present, it is resistant to insect pests and resistant to jacids. Really you can see the cotton, and resistant to leaf beetle you can see in the wheat, and high aspartic acid and also low nitrogen and also sugar. It is resistant to the maize and also stem borers. Next, biofortification. It is increasing the protein. It is increasing the main breeding crops with the higher protein. Breeding crops with a higher protein and healthier to improve the public health is called, it is a biofortification. So what are the main objectives? So it is increasing the protein content and quality and vitamin content and micronutrient and also mineral contents. Okay, so next IR giving the some of the crop varieties. So there are different components that can be present and also increase the nutrient quality, and also what will happen is you can see first vitamin E. Okay, vitamin E mainly you can see carrot and spinach and pumpkin, and vitamins is the bitter gourd and bathua and also tomato and mustard, and iron and also calcium mainly present in that spinach and also bathua, and also proteinaceous compound, it is the bean and also broad bean and garden pea and also French and Dolichos labulus, such as vegetables that can be contained in this. These are the proteinaceous components. Okay, next you can see the tissue culture. So tissue culture you can see, so artificially the production of new plant. So in lab condition, that's we call it is the tissue culture. Actually, what are the tissue culture steps are present? You can see the preparation of nutrient medium. Plants, how it is naturally absorbing the minerals. So macronutrients and micronutrients. So all these are in lab condition. That means in vitro condition we are prepared we are preparing the medium, that's we call it is preparation of nutrient medium. So this nutrient medium, so this nutrient medium prepare, after this nutrient medium it can be sterilized in a culture medium. So the sterilizer medium, it is sterilization in which condition means 21 degrees centigrade, okay, 21 degrees Celsius pressure at how many minutes means it is nearly it is 15 minutes, okay. So the sterilization culture medium 121 Celsius pressure, nearly about it is a 15 to 20 degree 20 minutes we can sterilize. So automatically what are the, whenever if you sterilize, what are the microbes are present, that the microbes are can be there, okay. After that we are next about the preparation of an explant. So any part of the plant, we call it is explant. So the explant is prepared from any part, means it is anthers and pollen grains and ovules and root, tip and root, stem, leaves, any parts from that will take this explant. So the explant size nearly about it is 0.4 to 0.4 into 0.4 mm size, okay. So next, incubation for this growth, what are the explant is prepared, okay. What is the explant is prepared, that is inoculated into this growth medium in the test tube. So in this process, inoculation process, we are using the operator, it is laminar airflow. So in the, whenever it is the presence of laminar airflow, the explant is inoculated into this culture medium tube. So how much of time it can be incubated means 3 to 4 weeks. Whenever it is 3 to 4 weeks, it can be incubated. So it can be formed the undifferentiated mass of tissue in the test tube. That's we call it is the callus. So the callus. So in the callus, so high oxygen and low cytokines, as a result, it is an increase in the root concentration. Okay, roots are it can be increases. So next it is the accumulation of plant and transfer to plants. Whenever this callus is formed, after that we are in the test tube, it can be added the some of the growth hormones. Whenever it is the growth hormones, it can be added. So again it is one more 3 to 4 weeks. Again 3 to 4 weeks, the plant will be developed in the test tube. So after that it can be separated into plants, okay. Whenever it is what are the natural condition, room temperature we have to maintain. Later the growth will be sufficient. With this plant it can be transferred into a plant or pots. So in the pots we are applying to the, what is this, it is the coconut powder, okay. The soil rate we can apply, so automatically plant it grow well, okay. This is the main tissue culture step, simply I am saying. So next of mine, what are the main applications? So micro propagation. So the thousands of plants within a short period of time, within a space, it can produce, and that's we call it as the micro propagation. So the meristem culture, the meristems are used, it can produce in various plants. Somaculus means it is the parent plant, how it is present, the clones are same as usual presence, that's we call it is a somaculum varieties. And somatic hybridation, so the fusion of two protoplasts, that's we call it is a somatic hybrids, okay. This is the main tissue culture applications. And single cell proteins, so what is meant by here the single cell proteins. Single cell proteins means, what is meant by here the single cell protein. Actually, single cell protein means the dried biomass of species, of microbe that can be used as a protein. The dried biomass of species, okay, of microbe that can be used as a protein in our diet. We call it is a single cell protein. So what are the examples of single cell proteins? Algae, that is a chlorella synedesmus. Yeast is the Saccharomyces cerevisiae, and Candida utilis, generally we call it is the torula yeast. And cyanobacteria, example it is the spirulina. Okay, cyanobacteria, you can see it is the spirulina. Sorry, cyanobacteria. Okay, the cyanobacteria. So and also fungi. The fungi is the Fusarium mulliforme, Fusarium mulliniarum, that's in fungi. And bacteria is a Methylophilus and Methylotrophus. So these are used. It is single cell proteins. Actually, it is the one cow. The cow, actually, how much is present? It means it is of 250 kgs. 250 kgs of cow. How much of it is the proteins are produced? Means it is the 200 grams of it is the protein is produced. Okay, and 250 grams of this Methylophilus, okay, bacteria, to produce nearly 25 tons of it is a protein. Very very important for me. 250 grams of these Microbes bacteria can produce now 25 tons of it is a proteins. Okay, so these are the strategies. You can see the objective bits.